Well, thank you, Nathan, for the introduction. And thank you all for coming here. This is a great symposium. I'm learning a lot. It's really fantastic. So I'm going to change gears a little bit, and I'm going to talk about a couple of different therapeutic platforms we are building in our lab to counter these emerging and evolving pathogens. The way we look at pathogens is, we've talked about heterogeneity, there's about hedging, there's variability. And in our labs, when we evolve our good old lab strains, we see that within a matter of days to weeks, we can generate tolerance to antibiotics. Now, that is crazy. And we begin to appreciate and understand how nature can quickly change and how organisms can come up with various different solutions to counter a certain situation. And so when we're thinking of therapy, we have to start considering how nature can evolve. And that's why we want to go back to the drawing board and start creating newer therapies on newer platforms. And in order to do that, in our lab, we end up using a whole range of interdisciplinary approaches, ranging from synthetic biology to systems biology, nanobiotechnology, and even synthetic chemistry, with the goal of designing antimicrobials rationally. And here today, I'm going to focus on towards combating the problem of drug-resistant pathogens. So as Mark very aptly put forth the problem of antimicrobial resistance, and it's definitely on the rise. So a couple of years back, the Centers for Disease Control released the report stating that nearly 2 million people are infected with drug-resistant infections in the US. Out of which, unfortunately, 23,000 people die every year. And that statistic was considered to be 700,000 at a global scale. Now, as Mark mentioned, the report that was generated a few years back by the UK government and the Wellcome Trust, it projects that in a matter of 30 years, if current trends continue, the fatality could go up tremendously and would exceed any other kind of other disease. And I guess the important point is to realize this thing is real. Whether you like it or not, we are approaching a post-antibiotic era, and we have to make serious changes in how we design therapy right now. And in order to do that, one has to appreciate the history of antibiotics. So if you go back and go back in time, you see that even before the first commercial antibiotic came out, penicillin, there was already a strain reported to be resistant to it. For tetracycline, it was released in 1950. By 1959, there was a strain reported to be resistant to it. And the story continues. Every single time an antibiotic is introduced in the market, within a matter of months to years, you see resistance. In fact, that's one of the major reasons also for a lot of companies to move away from antibiotic research is because you spend millions to billions of dollars building these antibiotics, and even within clinical trials, you end up seeing resistance. So it's a serious issue. And so the World Health Organization and the Centers for Disease Control have come up with their priority lists. So they have priority 1, priority 2, and so on pathogens. And because these pathogens are very drug resistant and they're spreading at a very fast pace at a global scale. And here I've highlighted one such family of pathogens called Enterobacter ceae that are a huge problem right now because they're highly drug resistant to almost all antibiotics, and they're very prevalent. And as you can see in this graph, they're kind of increasing at a very fast pace. And on the other hand, while these bugs are evolving, innovation is not catching up. So the new drugs that are being approved by FDA are declining. And even those that have been approved for the last 5 years, they are targeting gram-positives. And very few antibiotics are actually targeting new classes or have new targets per se. So we need to do something different to catch up with these bugs. And the way we look at, in our lab, this problem of antibiotic crisis, we think of it as an hourglass situation. So on top of the hourglass, you have new antibiotics that the bugs are sensitive to, or antibiotics that bugs are sensitive to. And at the bottom, you have antibiotics that the bugs will eventually gain resistance to. And it's just a matter of time till the antibiotics move from this top part to the bottom part. So the way we think of solutions for this particular problem is, one, developing new, and I emphasize smarter antibiotics. That is learning from what has happened over the last few decades, learning from how bugs are evolving and trying to understand if we could come up with strategies that could counter those processes. and creating smarter antibiotics in the process. And the second thing that is an interesting thing, it's rather a new approach, which is how can we constrain this part of the hourglass so that either we can stop or, in more realistic situation, probably slow down the process in which bugs are gaining resistance or gaining tolerance to antibiotics. So in other words, can we slow down the rate of adaptation or evolution of bugs to antibiotics? And so today I'm going to be talking about 3 different platforms that we are trying to develop in our lab to approach or to address these 2 solutions. So 2 platforms. So one I call the FAST platform, other one is a quantum dot-based platform, address solution number 1. And there's another platform we call CHAOS that addresses solution number 2. So the first platform, which is called Facile Accelerated Specific Therapeutic, or FAST, is a platform that will use antisense antimicrobials to target non-traditional pathways and genes in order to eliminate multidrug-resistant bacteria. And the reason I like to call this platform FAST, because this platform has a very ambitious goal, which is that we want to go from drug discovery to testing in a matter of hours to days, compared to conventional months to years. And that's an ambitious goal, but I do think that as we are facing newer strains of bacteria and viruses, there was a lot of discussion yesterday about tracking in real time virus outbreaks. I think we also have to prepare, on the other hand, therapeutics that can counter those strains in real time. And this also addresses the huge need for personalized medicine as well. So let me just quickly remind what's being done as of now. So the conventional antibiotic discovery strategy still relies a lot on small molecule screening. And when I say small molecule or say natural product screening, we're essentially borrowing from nature. So we're borrowing from bacteria, fungi mainly, and we are basically screening thousands to millions of molecules for targeting a certain protein that we isolate from a certain pathogen. And while this process is very well established, there are certain concerns with it. So as you can imagine, this is something that is highly tedious, requires skill, labor, time, and as they say, you're not paying for the drug that worked, you're paying for the drugs for the other things that didn't work. So it again is something that needs a lot of investment. So, but I have another concern with this strategy, which is something that we see in our lab strains, is that we are actually playing to bacteria's strength. Bacteria has been doing this for millions of years. They've been using small molecules to kill other bugs so that they have a competitive edge in a certain environmental niche. So if we are kind of borrowing those molecules or even modifying those molecules, I believe bacteria have various ways to counter those kind of situations. So the way we see it is that it's a short-term solution. Now, recently, scientists have started investigating more pathogen-specific therapies. So in the last few years and decades, there has been a lot of study on the microbiome, and people have started understanding that protecting the microbiome is very important for our health. And so we want to create antibiotics that are targeting just the pathogen, but not necessarily hurting the microbiome. So in that respect, there has been a lot of excitement about CRISPR-based antimicrobials. So CRISPR is a platform for now being used a lot for genome engineering, but it was originally borrowed from bacteria. So bacteria uses the system to kill bacterial viruses or phages. And it essentially has a protein and an RNA called guide RNA that essentially recruits this protein where it goes and cuts whatever sequence you want to target. Now, this is great in the sense that it allows you to rationally create therapy. But on the other side, we again have issues of tedious cloning. Then again, you may not have a perfect CRISPR system for every pathogen. And when you're trying to translate things from lab to clinical strains, it is a pain to kind of move plasmids into the clinical strain. And again, we are plagued with the issues of inefficient delivery. So in our lab, we want to take advantage of this rational approach where we're targeting a particular sequence, targeting a particular pathogen, but at the same time, we want to remove any cloning. So we are using an antisense technology platform, and specifically, we are using a molecule called peptide nucleic acid, or PNA. And this PNA is essentially a peptide. So it's a peptide backbone, and the side groups are basically nucleobases. And it's a synthetic molecule that's been designed to have perfect spacing so that it mimics naturally occurring nucleic acids. And because of its neutral backbone, it actually has higher binding affinity to RNA and DNA compared to naturally occurring RNA and DNA. Now, we can design these molecules to target any gene we want and inhibit the translation of the mRNAs. And the great thing is we don't have to do any cloning. So it's a very facile approach to create therapy. But obviously, there are also some limitations, and the limitations include the transport isn't as good. It's inefficient when compared to small molecules, and that's an area that our lab is actively working on. And also, compared to other therapeutics, the bioavailability is low, and again, that's an area we're working on. So, but the great thing with something like this is that you suddenly can target a lot of the different things that you couldn't before. So conventional antibiotics typically target these 3 pathways in bacteria. So if you basically look at the various pathways in which, where the essential genes of E. coli are, the pathways that I highlight in red are the ones that there exists at least one antibiotic targeting the pathways. But if you see these other additional pathways in black, they have never been targeted using conventional antibiotics. And if you look at the classification of essential genes by pathways, you see there are a lot more genes and pathways available for us to target. In fact, hundreds of them. And a technology like PNA can actually achieve targeting those pathways. And the way it works is you design a 12-mer PNA to target the 5' untranslated region, and more specifically, what works best is if you target the start codon region of a messenger RNA. And the idea is that we want to block binding or migration of ribosome on the mRNA, and in that way, we prevent translation of the mRNA. Now, PNA is highly specific, and it can, even like a single or 2 base pair mutation will cause loss of binding affinity, but that's not a problem. Why? Because ultimately, we want to use a cocktail therapy wherein you're not targeting one gene, but multiple genes. And you can actually do a simple math to figure out how many PNAs do you need so that your mutation rate actually, or the ability of bacteria to mutate at all sites almost goes to 0. But the long story, the main point here is that a technology like this is highly flexible, and you can rationally design therapy to target any gene you want. So in initial studies, we actually designed PNAs to target resistance genes in bacteria. So the idea was you target resistance genes and resensitize the bacteria to the original antibiotic. So here I'm showing you the 5' UTR region of a messenger RNA from TEM1 beta-lactamase gene. And I'm just showing as an example 3 sites here that we targeted. And what we see is that if you target the start codon region, it seems to have the lowest minimum inhibitory concentration, or MIC. So in this case, it turns out that at around 2.5 micromolar, the bacteria is sensitized to the antibiotic. If you target the ribosome binding site, we actually had a 10-fold higher MIC, and if we targeted another conserved RNA structural unit downstream, we did not see, in a working concentration range, we did not observe a MIC. So, and then we have done this for multiple genes, and what we find is that start codon indeed is the best site to target. So once we know that, we can now target a whole lot of different genes and pathways and essentially increase our antibiotic portfolio. So we can design PNA to target the start site. We can also design PNAs that have homology across various pathogens. So you can actually customize which combinations of pathogens you want to target. So here I'm showing an example where we started off with essential genes in E. coli, and we built this bioinformatics toolbox because there isn't one for PNAs. And so now we can download genomes of any pathogen of interest, design PNAs for any pathogen of interest. We can also look for internal off-targets with 1 or 2 base pair mismatch. We can cross-list across pathogens and basically have PNAs that target 1 or more pathogens. So here, what I'm showing is that you can basically narrow down the number of targets, including thermodynamic constraints on the PNA. And in this example, let's say eventually we ended up getting 71 PNA targets across various pathways and various non-traditional pathways. And even if we look at a very small fraction of that, even if we sample like less than 10% of that library and design PNAs that target non-traditional pathways such as, let's say, FOLC, which targets metabolism, FFH that targets signal recognition, LEXA that targets stress response, or something that targets traditional pathways like gyrase that targets transcription. However, that's a new gene that we are targeting, or RPST that's targeting translation. We then design these PNAs, supply directly in culture, no cloning, and then see what happens. And then we treat clinical isolates with these PNAs. And the reason we do that is because ultimately you have to see whether these therapies work against these highly drug-resistant pathogens that affect patients. So we have collaborators at the medical campus at University of Colorado who supply us with a lot of drug-resistant pathogens. And again, I'm showing you here an example, a set of pathogens. So we have 2 E. coli strains. One of them is carbapenem-resistant E. coli, which is a last resort antibiotic, so it's resistant to that. Other one is the generic MDR E. coli. Then we have 2 strains of Klebsiella pneumoniae. One has extended spectrum beta-lactamase, and another one has the notorious New Delhi metallo-beta-lactamase. Then we have some generic MDR Salmonella. Here I'm showing antibiotics, and this is the phenotypic characterization. R or anything in red indicates the pathogen was resistant, was growing at concentrations higher than the clinical breakpoint. S is sensitive, I is intermediate resistance. And we also sequence these pathogens to have a correlation between the phenotype and the genotype using existing databases. And for the most part, we actually have a pretty good correlation between the resistance phenotype and the genotype. But sometimes we have surprising things where we don't find any known resistance gene for the resistance phenotype, which, by the way, I'm not going to talk about it today, but also makes a lot of connections to the other work we do in our lab, where we find a lot of non-genetic reasons for antibiotic tolerance phenotype. So the interesting thing is that when you treat these highly drug-resistant pathogens, and the reason I say this highly drug-resistant because many of these pathogens are actually growing at concentrations of antibiotics 1,000 times higher than the clinical breakpoints, which is insane. They are almost thriving in antibiotics. But despite that, they're actually pretty sensitive to the PNAs that we have designed for them. So if you design PNA that targets a scrambled sequence that doesn't exist in the bacteria, you don't see any toxicity with the bugs. But for our PNAs, you see quite a bit of toxicity. And if you look at the treatment, so for 70% of the treatments for the various PNAs, we observe significant growth inhibition. Around 54% of our treatments show 50% or more growth inhibition, and around 27% of our treatments show greater than 95% growth inhibition. Now, if you compare that with small molecule-based approaches, success rate typically is 0.0001%. So what it shows that rational design of therapy could actually be very useful in treating these highly drug-resistant pathogens. And importantly, diversifying the pathways, targeting these non-traditional pathways and genes may actually be very useful in perturbing these pathogens. Now, if you take the PNAs that didn't work as well in monotherapy or worked partially and combine them with traditional antibiotics, we see something really interesting. We see a strong synergy. So it seems that the PNAs can also potentiate antibiotic activity. And what is very interesting is that these pathogens actually had the resistance genes for these antibiotics. And despite having those resistance genes, the PNAs are able to potentiate activity of the antibiotics, which means that for the resistance genes to manifest their function, they are interacting with the host metabolism or cell signaling, and by perturbing some other additional pathways, you may actually be able to prevent the resistance gene from function at all. So our FAST, or the PNA platform, indicates that there can be strong therapeutic potential if you rational design therapy. So what's next? So the next step is to actually do this at a larger scale and systematically across pathogens. So we are building the bioinformatics tool to do that. And the next step is actually to also improve the delivery of PNA. And I wouldn't go into the details of this, but by doing the bioinformatics, we can actually design PNAs for any desired pathogen. We can also look for off-target effects in gut microbiome. So here I'm showing representative gut bacteria, and we have a longer list that we're working on. And what you can do is you can customize therapy. So let's say in the mix of E. coli, Salmonella, and Klebsiella, let's throw Staph aureus as well. That's a gram-positive. And we can design a PNA that targets each one of these individually. We can design a PNA that targets 2 of these pathogens at a time. We can design PNA that can target 3 at a time or 4 at a time. And what I'm trying to indicate is that as the future of therapy, you can actually even customize how you would like to create therapy, customize antibiotic treatment for various patients. Now, as I mentioned, transport is still a challenge, and in our lab, we are using various approaches. One of those approaches is synthetic biology, where we are trying to learn from what nature does. So bacteria uses various kinds of secretion system, and one of those secretions is the type 3 secretion system, where bacteria express proteins that allow them to dock on mammalian cells. Then they express a needle and inject proteins in the mammalian cells, and eventually they can also intracellularize within mammalian cells. So we are trying to repurpose or hijack this type 3 secretion system and in fact use it for delivery of PNA. So essentially we're using bacteria as an agent for delivering our PNA. And by doing that, we are also kind of integrating a kill switch. So once the bacteria is intracellularized, the kill switch is turned on, and the bacteria is lysed to release the PNA for treating intracellular infections. So yesterday, we heard a lot of talks on tuberculosis, and that's an agent that invades a lot of mammalian cells and resides in mammalian cells. But there are a lot of other bacteria, such as Salmonella, that do the same thing, that can infect mammalian cells, live there happily, evade immune response, and spread around the body. So here I'm showing an intracellular infection of Salmonella with macrophage cells and osteoblast cells. And the green indicates Salmonella expressing the green fluorescent protein. And as you can see that when we treat this infection with PNAs that are transported using the type 3 secretion system, we can observe pretty high clearance, up to 90%. And we are now pushing the boundaries with this particular approach. Something that I'm not going to talk about today, but we're also trying to learn from what's been done for peptide-based drugs, and we're trying to use synthetic chemistry to also improve the transport of these molecules. So eventually, the goal is to kind of make this into a seamless process, so what I like to call a fast platform, where we design, build, and test these therapies very quickly. So our informatics toolbox will give you information within a matter of minutes what should be your list of PNA targets with probabilities associated with each of them. We have actually now started synthesizing a therapeutic on site. So the idea is eventually it's an automated process where the computer tells you what therapy should work. Then that moves on to an instrument that designs the therapy on site. Then we're building our transport systems, and then we have already established a bunch of infection models where we're going to test our therapies in a matter of a day. So eventually, the goal is that this whole process will be done in a matter of hours to days. So the next is, OK, so we have designed therapy. Now, can we slow down evolution? So in order to do that, we have built a platform that we like to call CHAOS, and it stands for Controlled Hindrance of Adaptation of Organisms. And the idea here is that we want to slow down evolution of bacteria. And in order to do that, we need to understand how bugs evolve. So many years back, somewhere in the 1930s, Wright came up with this shifting balance model of evolution, essentially basically describing how bacteria evolve or move along a landscape called the fitness landscape. So in a very simplified world, if we think bacteria only has 2 genes, gene A and B, if bacteria is in this wild-type state and it sees the greener pasture, which is a higher fitness state, it would like to go there. And in order to do that, bacteria will, in the conventional sense, mutate gene A and B, and at a population scale where you have thousands or millions of bacteria, each individual bacteria is trying to figure out a solution, and eventually some of them will figure out a solution and traverse this landscape. Now the question is, what allows them to traverse this landscape? So it's a phenomena called epistasis. So as Mark was talking about epistasis, so here, epistasis, let's see what epistasis is. So let's say we have wild-type bacteria. Let's say it has a relative fitness of 1. Now let's consider a mutant A. Let's say its fitness is 0.9. Mutant B, its fitness is 0.92. Now if we combine those 2 mutants, and it turns out the fitness is 0.81, then according to this multiplicative model, epistasis, which is the 0.81's product of these 2, net epistasis is 0. However, if the fitness is greater than 0.81, then we say it's positive epistasis. If it's less than 0.81, it's negative epistasis. And positive epistasis is basically pushing the bug towards more evolvability, and negative epistasis means less evolvability. And in our lab, we are actually using these two approaches for very different applications. Less evolvability, we are pushing towards therapy. More evolvability, we're pushing towards metabolic engineering. But the question is, all of this is defined in the mutational kind of landscape, but mutations cannot be induced in a clinical setting. So is there a way of inducing negative epistasis, but not at a mutation scale? And in order to do that, we turn our attention to gene expression. So our hypothesis is multiple gene expression perturbations will induce negative epistasis. So we kind of like to think of our chaos platform like a hammerhead game. So if any of you have played the hammerhead game, so where you have a bunch of, let's say, squirrels popping up their heads, and you're given a hammer to hit the squirrel on the head. And you win the game when you hit all the squirrels on the head. Now, if you're given just one hammer, the chances of success are less. But if you're given, let's say, 3 hammers, the chances of success are high. And that's how we see nature when nature is constantly evolving. These fitness landscapes are dynamic. And there's always nature is going to find a solution. So in order to have robust therapy, maybe we need not just one, but multiple hammers to hit multiple points or multiple nodes in a network to increase our chances of success. And we want to do this thing so that we can artificially induce negative epistasis, and ultimately the goal is that this fitness barrier that the bacteria needs to cross to get to the evolved state, we somehow increase it enough that it is not able to cross that barrier. And so our hypothesis becomes now multiple gene perturbations will induce negative epistasis, and such induced negative epistasis will enable slowing down of bacterial adaptation. So in order to do that, we have built a whole range of CRISPR constructs in our lab. So we use the deactivated CRISPR system that can either inhibit or increase gene expression. Again, it's very rational. We know what we want to target, and we can controllably increase or decrease expression. In the interest of time, I'm just going to talk about 2 examples. So one is a set of stress response genes, and one is a set of essential genes that we targeted. And we created all possible combinations of perturbations of single, double, triple, and quadruple perturbations within each of these sets. And then we took all of these constructs and competed them with a control strain in the presence of an antibiotic and saw what happened. And what we find was something really interesting, that as you increase the number of perturbations, you start seeing decreases in fitness. So the more reddish hue you have, lower fitness. So the perturbed strain is less fit than the control strain. And when you do the math to calculate the epistasis, it turns out that as increasing the number of perturbations, your negative epistasis is increasing. And in fact, when we create the stress response quadruple perturbation, the essential gene quadruple perturbation, and actually a combination of 2 stress response genes and 2 essential genes, we actually see a sharp increase in negative epistasis when we're targeting essential genes or some combination of essential genes. So obvious next thing was to see, adapt them and see what happens, whether we can slow down evolution. So in a short-term adaptation, we adapted these constructs in presence of ciprofloxacin selection pressure, and we measured the increases in the minimum inhibitory concentration to ciprofloxacin over time. So this is the control strain and how it adapts. But if you look at the single gene perturbations, you see generally a slightly upward trend. And that upward trend seems to be stronger with a higher Pearson correlation coefficient compared to, say, the essential genes. But interesting stuff happens when you start looking at those quadruple perturbations, especially the ones that showed high negative epistasis. So here we see that when you look at the stress response gene quadruple perturbation, there is still a positive trend, but it seems to be weaker compared to the individual stress response perturbations. When you look at that quadruple perturbation, which combined 2 stress response and 2 essential genes which had high negative epistasis, you see flattening of the MIC profile, which indicates the bacteria is not able to adapt in this given time frame. And when we looked at the essential gene quadruple perturbation, because the essential gene perturbations were also had a little bit of a therapeutic effect, we saw a decline in the MIC. And when we removed the replicates that were dying out, we actually saw a flattening of the MIC profile. So what it indicates is that if we understand the epistatic interactions between the various genes we have, we may be able to actually figure out combinations that will lead to negative epistasis. And then we can design therapies that can actually target those genes and induce negative epistasis at a clinical setting. And now we are really doing this at a systematic scale where we're trying to find all combinations possible at a transcriptome level to isolate the gene combinations. And then finally, I'd like to finish up with a third platform, which is a quantum dot-based antibiotic. And it's a very interesting one because it's targeting a very different pathway, redox homeostasis. So this is very different in the sense that here we actually have a semiconductor material as an antibiotic. And this material has been engineered in a way that has very precise conduction valence bands with precise band gap. And the whole idea here is that we want to excite this quantum dot with a certain stimuli. We're using light right now, to produce electrons with very specific energies. That electron then goes and reacts with dissolved molecular oxygen to produce a very specific oxidative stress radical, here superoxide. That superoxide then goes, reacts with various other things inside the bacteria to eventually kill it. So these are very tiny 3 nanometer particles. So unlike PNA, I don't have to worry about transport that much here. Once they're in the cell, we have the stimuli that produces the electron and finally the superoxide. When bacteria sees the superoxide, and we can actually tune the flux of superoxide by increasing the intensity of the light or by changing the concentrations. And by the way, we're working at nanomolar concentrations with these quantum dots. So 10 nanomolar or so is enough to kill the bacteria. But for mammalian cells, the threshold for handling that superoxide stress is much higher. Because remember, macrophages use superoxide and nitric oxide to kill pathogens. And here we are in some ways thinking about it, learning from macrophages and aiding macrophages to increase the superoxide flux for the pathogens. So the way the superoxide works is that it uses the Fenton chemistry, and it can also basically plug into various points in the redox pathways to kind of perturb or break down redox homeostasis. And we have confirmed using electron paramagnetic resonance and also genetic screens that the radical being produced is superoxide, and it is not a material-dependent effect. So that's one of the things people think of when we talk about quantum dots. And this particular quantum dot I'm talking about right now is made of cadmium telluride, so that is one big concern people always have. That's maybe because of the material. But that's not the case, because here, for example, I'm showing that when you only excite the quantum dot with light, do you see this dye glow, which this dye is an indicator of oxidative stress, but in dark, you don't see cells glow that much. We also have a bunch of various controls to prove this point. So this particular antibiotic works via bactericidal mechanism. It's fantastic. It kills a whole range of bacteria, not just gram-negatives, also gram-positives. And when we create a library of quantum dots, so here I'm just showing this set where you still use CDTE, but you shift the conduction of the valence band or the band gap of the quantum dot. So you create CDTE 2.3 and 2.2. You see that these two quantum dots don't have therapeutic effect. So this is not a material-dependent effect. This is totally a function of the electronic property and the fact that we are producing electrons with very specific energies that produce superoxides. Also, when you use the same band gap, but you use another material, say CDSC, you still don't see the therapeutic effect. That's again because of the fact that now you may have the same band gap, but you're changing the oxidation and reduction potential of the electron being produced, so of the radicals being produced by the quantum dot, and that doesn't work. So it's the precise superoxide that's causing the therapeutic effect. And this is actually very good at even potentiating antibiotic activity. So we have a whole range of pathogens, and here I'm showing their GIC50, and we see that a bunch of pathogens, even at 50% growth inhibition, are growing at concentration 1,000 times higher than the clinical breakpoint. So the red line here indicates the resistance breakpoint, blue line indicates sensitive breakpoint. So anything above the red line is resistant, anything between the lines is intermediate resistance. And a bunch of them, as you can see, are pretty resistant. But when you start adding the quantum dot, increasing the concentration, you see that you can drag all of those pathogens down below the sensitive line. So this is another something interesting because it's telling you that despite the resistance gene, you are potentiating antibiotic activity, which means that if you target at other pathways, like we did with the PNA, or you're perturbing redox homeostasis, then you can sensitize antibiotics. Another great thing about putting oxidative stress is you have to wake up cells. So those who deal with persisters have this challenge that cells can basically control their metabolism to kind of wait till the stress goes away. And when you throw oxidative stress, you are kind of forcing the cells to wake up and counter that stress, and that automatically makes them sensitive to the antibiotic treatment. So, and this is again great for treating intracellular infections because transport is not a challenge. So we have actually treated in salmonella infections in various cell lines. It works great as a monotherapy as well as a potentiator. We've also done in vivo work in C. elegans, and one of our next steps is to move into murine models. We've also started working with Lyme disease to explore whether quantum dots could be used to treat intracellular infections of Borrelia. So with that, I'd like to conclude, and hopefully I've shown that if we diversify our strategies, and in the first case, if we use a platform like FAST that is nucleic acid specific and can target various other genes and pathways, we can actually eliminate or sensitize very drug-resistant clinical isolates. By perturbing various genes, we can also induce chaos and control the rate of evolution. And as you can imagine, these two platforms can be interfaced pretty easily to translate the idea of chaos in the clinical setting. Then I also showed a third platform with the quantum dot, which is targeting redox homeostasis and can, again, eliminate highly pathogenic bacteria. And ultimately, I go back to the original hourglass slide, and the whole idea is to create smarter antibiotics that can be rationally designed, that are adaptable. So if the pathogen evolves, we should be able to change our therapy quickly, scalable, easily manufactured. With that, I'd like to thank all my students who participated in the work I presented today, my collaborators who participated in the work I presented today, Prashant, Nancy, Corey, Tarun, and Bob, and funding agencies, and thank you guys for being patient with me. Thank you.
I'm a non-professional in this area, so if this is obvious, tell me. But when you talked about the migration of an organism to a desirable new mutation, and then you described a process for interfering with that migration, what is the gradient? You used the word like, it likes to go to this new place. Is there a gradient of some sort that it's following that you're interfering with? I didn't catch what the motivation for moving is and how you target what to interfere with.
So I'm guessing you're talking about the chaos platform, am I right? So the evolution, the one with the fitness landscape?
Yes.
So basically, here we're trying to understand how bugs traverse this landscape.
Yes.
And I guess there has been discussion about heterogeneity, and we have to understand that when we are dealing with bugs, we're not just dealing with one bug, we're dealing with, say, hundreds, millions, depending on the context, lots of bugs. And each individual bug is basically trying to figure out how to get to a bit higher fitness state. Or if there is some sort of antibiotic stress, they want to survive and reach that fitness state.
So if they're feeling less stress, there's a gradient of less stress that's giving them a direction?
No, in this case, we are not talking about less stress. In this case, we are talking about how they are basically making changes either at the genome scale or in our case at the transcriptome scale so that they can tolerate that stress. So they are basically changing some sort of expression level of the genes such that they get to a state where they can tolerate the antibiotic. And as long as they can tolerate for some time, eventually that will manifest into some sort of a resistance gene in the long term. So what we are trying to do is we're trying to understand this very fundamental process of how they are getting there, and we're trying to interfere with it and stop that process in the beginning.
Right. And my curiosity is, as I heard what you just said, there's an internal sensitivity to a disintegrating process, and they move away from that disintegration. The rise of stress is some kind of deterioration that it senses and can move away from.
Yeah, and that's innate in all of our cells, I mean, not just bacteria, is that whenever there is a stress, cells will respond. And the fastest and the quickest short-term response is changing their gene expression. And when you're sampling a population, some of them will come up with the correct response, and some of them will with the wrong response. And those will get eliminated from the population. And the correct ones are the ones that persist long enough eventually to repopulate.
So would you say that?
Can we move on to the next question? Let's continue that discussion in the panel discussion. Let's take one more question here because we're already over time.
Okay, a quick question. So for this PNA, how difficult is to synthesize it, or is this commercially available too?
Yes, it is commercially available, and it is still a little bit, it's expensive on commercially, like if you buy them. It's not too difficult to synthesize because, again, remember the backbone is peptide-based, so you can use solid phase chemistry, and that's what we're doing in our lab. So it's fairly easy to synthesize them.
Okay, let's thank the speaker again.